Hello, thank you very much. So I'm going to present what we do at IKTOS. We really focus on early stage drug discovery and drug design, which we like to compare to polishing a diamond with some metrics that have been presented by the previous presenters in terms of global duration of the process, costs of the main three stages: hit discovery, hit to lead, lead optimization, and success rates, which basically is appallingly low, and so there's a lot of room for improvement. So the question is, can big data and AI help? Of course, we're here because we believe it is possible. So what do we mean by AI for drug discovery? Actually, it's a very, very large field and domain, and many companies are doing many different things, more in the field of biology, or compound profiling, deconvolution of phenotypic assay data, drug repositioning, activity prediction, protein-ligand interaction prediction. We really focus on de novo drug design using generative models. So who are we? We are a very young startup company, founded in September 2016, at the very beginning of our history. We are a team of 15 people now, combining skills in chemo-informatics, computational chemistry, medchem, software development, machine learning, data science, and IT. We have developed and are still developing a proprietary technology for automatic drug design using deep generative models. And we have already several references with pharma clients as a service provider, but we are also developing a software platform, a SaaS platform, because we ambition to put the technology in the hands of the medicinal chemists. These are the pharmaceutical companies for which we have announced collaborations with. The latest one was Orion, no later than yesterday. So we like to compare medicinal chemistry, and in particular focusing on the challenge of multi-parametric optimization to a Rubik's cube. It's quite easy to solve one face of the cube. But when you want to you make two faces or even more, 6 faces, you have to break the first face. So if you have an active molecule, but it's not soluble, if you increase the solubility, you will lose the activity. And the real challenge of the medicinal chemist is really, one of the real challenges of the medicinal chemist is to be able to find the solution of the cube in a reasonable number of steps, because each step is much longer and more expensive than, of course, any move you can do with the Rubik's cube. So, of course, the chemical space being what it is, absolutely infinite, the solution should exist somewhere. The question is, where is it? Where can we find it? And and there are many tools that have been developed and technologies that have been developed over the years to predict the activity of a molecule. Provided you have the structure of the molecule, that you have enough data, or you have the structure of the target, you can have developed in silico tools which will tell you with a reasonable level of performance whether or not your molecule will be active on the target. So the real question is that this tool cannot do the other way around. They cannot tell you which molecule you should do in order to have all your objectives set at the same time. So when we started the company, it was really deep learning, and had already started doing a lot of amazing things in many fields, like being able to colorize automatically black and white images, or to be able to generate text describing the image automatically. Of course, the famous example of Go games and those cats, which are automatically generated by the system. So our idea was to say, okay, why not try to generate molecules instead of cats. If we can train algorithms to generate new text or music or images, why not generate molecules? And at the time, that was in 2015, deep learning in chemistry was really at its infancy. The first academic work to be published was actually the Merck-Kaggle challenge in 2012, where they demonstrated that multitask learning using deep learning for prediction purpose enabled to increase the level of performance compared to standard QSAR models. Then they decided to replace the classic Morgan fingerprints by features which would be generated by the computer itself from the chemical structure of the molecules. That was in 2015. And then only in 2016, late in October 2016, just 1 month before we had created the company, was the first paper by Duvneau and Gomez-Bombarelli published on the use of variational autoencoders to generate new molecules using deep learning. And since then, it has been a race, and there have been tons of papers published with different algorithms. Academic groups are very prolific in this space, and there was a nice review in February 2018, The Rise of Deep Learning in Drug Discovery, which gives a good overview of what's happening in the field. So when you look at what's been demonstrated right now, now it's very easy to get some code of generative model available on GitHub, run it at your computer and start generating molecules, but it's not enough to solve real-life problems. One of the early issues was basically the quality of the molecules that was generated, drug likeness, stability, chemical relevancy of the molecules that have been pretty much solved. Now, synthetic accessibility has also been, many solutions have been developed, especially in the field of retrosynthesis and synthetic complexity score. So now you can generate molecules which will be synthetically accessible with generative models. Right now it's possible, and it's been demonstrated by several papers in silico, but also in vitro with molecules which have been generated de novo and measured on one single objectives or two objectives to be able to generate molecules which will meet this. However, nothing yet has been published on optimizing on more than 5 objectives using deep generative models. Some other issues which have been described is the tendency of those algorithms to fall into potential wells. All the molecules which are generated will more or less look alike, and that's called the mode collapse. It's a critical issue in optimization in machine learning, and also generating new scaffolds and 3D approaches with generative models remain challenging. So what we do at Iktos is we use an approach where we combine a molecule generator which has been trained on a public data set of millions of molecules, and we combine that with a reinforcement learning process, which is basically where the reinforcement policy gives a score on molecules, which is provided by any kind of metrics that we decide to use to decide whether a molecule is good or not. And we rely usually on QSAR models, which are based on the data sets of our clients. So when basically that's the way it works, we generate one molecule. The molecule is scored by the QSAR model. The score is fed back into the system, and this information basically enables the deep learning generative models to decide in which direction of the chemical space to go to generate at the next generation a molecule which will be improved compared to the previous generation. And this reinforcement process enables to converge very quickly to molecules which maximize the scores which we are looking to maximize, and which is not a single score, it's multi-parametric optimization, and we get basically molecules which in silico meet all the criteria that we have set for the project: activity, specificity, solubility, stability, etc. So what does it look like in a real life project? So that was actually the first client we worked with, Laboratoire Servier. They were working at a project which was a late stages optimization project with almost 900 molecules, which had been measured on 11 assays. The primary activity was a complex ex vivo phenotypic assay, very expensive. There were 5 selectivity objectives on 5-HT2A to B, alpha-1, D1, sodium channels. There was a toxicity hERG assay, and there were 4 ADME assays: microsomal stability on human and rats, and absorption and efflux on CACO2. So very, very standard multi-parameter optimization challenge in a late-stage drug discovery assays. And of course, none of the molecules they had done was meeting all the 11 objectives. If you look at the data, you see that at the beginning of the project, for example, they were hitting 60% of the molecules were active. Then if you look at the next 50 molecules and then the last 50 molecules, which are in dark gray, they lost a little on activity. On other targets like sodium channels or hERG, they increased very much the performance also on microsomal stability. But on the other hand, they had very important selectivity issues on 5-HT2A, 5-HT2B, alpha-1 or D1. So they were not able to find molecules which were meeting simultaneously all objectives at the same time, and that's a very classic issue. This was the best molecule that they had made. As you can see, it was good on 9 objectives. It was borderline on the absorption, but it had still a very important efflux assays. And what's interesting is that on the molecules which were made, 80% of the last 50 molecules had this scaffold, this moiety, and it was there also in 60% of the data set. So it was the main moiety they had made their bets on during the whole process. So we applied our molecule generator as with the reinforcement learning process I explained. So if you see molecule is generated, it is scored on all independent QSAR models we had built for all assays. You have a result, and then it generates molecules, and progressively it gets molecules which are green on all targets. So these are the experiment results we got with the molecules which were made and tested. 11 molecules out of 150 which were meeting all objectives in silico. And as you see, we got a dramatic increase in performance with 100% of the molecules we made being good on all selectivity targets, 10 out of 11 of the molecules being good on hERG, on absorption and in efflux, a reasonably good performance on activity, only a decrease in stability, but still at a quite acceptable level. And what is more interesting is that when you look at the multi-parameter optimization, they had tested 48 molecules on all assays with an average of 6.4 objectives met, and our 11 molecules got an average of 9.5. So the increase in performance was really dramatic. And what's very important to say is that this takes 48 hours of computation. So it's a very, very, very quick process. If you have the data to build good QSAR models, the deep generative models are able to generate optimal molecules in a matter of hours. So what's more interesting is that, even more interesting is that in the 11 molecules we had 3 active molecules meeting all objectives, and this was the best one. meeting, as you see, all objectives at the same time. And what's interesting, if you look at this molecule, is that this scaffold here it was present only 6 times in the initial data set. So it was not completely new. They had tested it a couple times, but every time it was bad on efflux and permeability, so they had put it away. But the algorithm, which had no bias, which explores without any preconceptions, identified that with the right modifications, this scaffold could be useful and generate an optimal molecule. Also by adding some substitutions here on the right side, which had never been tested before in the initial data set. Another interesting look is that when you look at, we had computed here a principal component analysis to visualize the chemical space of the initial data set, and this is where basically all the best molecules were, the molecules that were meeting most objectives and also with a presence of pharmacological activity. So you might think that this is where the algorithm would generate the optimal compounds, but in fact it did not. In blue you see the molecules which have been generated by the algorithms. They are in another place, and the molecules which were the best ones were here. So what does it tell us? It tells us that when a chemist thinks that if he has a great molecule which is almost perfect, which only misses one of the objectives, he will always try to modify this near perfect molecule to find the optimal one. But it may be in a very different place, actually, that the solution to the problem lies. Because of the potential wealth, because the chemical space, it's not something which is linear, you need to go for potential different areas, and that's where the AI system has the capability, as I said, to explore without any preconception, just based on mathematical optimization. Another interesting graph is this one, where we looked at the property forecast index, which is a drug likeness index. It shouldn't be too high. It should be lower than 6. That was a GSK publication. This is a molecular weight, and in the size of the rings are the sp3 fraction, which should be as high as possible. And although this was not desired, it was not put in the optimization process. The molecules that were generated by the algorithms were at the same time, in an optimal chemical space with regards to those 3 characteristics. So on this case, we were able to generate optimal in silico propositions in only a few hours. And I want to stress the difference with combinatorial approaches, where the chemist needs to think a lot about what needs to be done, what are the scaffolds which need to be put as main choices, what kind of substitutions can be made in order to generate virtual libraries. This, in this case, is performed by the generative model itself, and in result, the exploration is exhaustive and very fast. We see it as an approach which is complementary to medicinal chemist propositions. In this case, we propose 150 molecules, but the chemists selected the ones which looked better to them. And we still believe that it's important to have the role of the chemist to examine the propositions. We may not be yet at the stage where decision could be made purely by the machine. We think that what we are going to do with Servier and with other customers is use this technology at a much earlier stage of research. Not start until the project is 10 years old or 5 years old and we have thousands of molecules, but start early on to guide the process along the way and to make sure that all compounds which are made will be optimal from a mathematical standpoint in order to reduce the timelines. And we believe also that there would be a high benefit of integrated in-house generic models in the process, again, to fuel this application of this technology at earlier stage of research. So, future impact of drug discovery. To conclude, we have similar ambitions as the one that were presented before. We believe that the potential of these technologies to increase the productivity of drug discovery is enormous. A few things we are working on. We are already working at generating new hits and new leads without any existing data on the process, but using the crystal structure of the target to generate new hits, new leads that will maximize the interactions between the protein and the ligand, and at the same time have optimal physicochemical and structural characteristics in order to reduce the timeline and the cost of the process. We also do work on scaffold hopping, working on public data, patent data, for example, in order to identify new chemotypes and generate new molecules with new chemotypes, building on QSAR models built on diverse data. We have also implemented an automatic retrosynthetic analysis technology, which can be coupled to the system to make sure that all the compounds we generate will be synthetically accessible, and we can propose synthetic schemes for those compounds. And finally, we also start working on hit-to-lead acceleration, working from HTS data in order to increase the speed to identify leads. So that's it. I will take your questions.
Hi, yeah, thank you. And you brought up a couple of interesting points. So I was hanging on to a question in the last few talks. Now might be a good time to ask it. First, just to understand correctly, I mean, you're building some sort of latent space from which you generate molecules in this latent space. Pre-question might be how you're forming this space, right? I assume some combination of, chemical properties or phenotypic assays or something to form some sort of space. The second would be when your question on mode collapse, right? So chemical space is huge. How many of these molecules you generate? I mean, how much of chemical space are we not able to generate from these latent spaces that we're typically generating now? Can we actually, how many are we not visiting? Can we at least get a picture for how many we're not, the gaps in chemical space that we can't generate from, which this may be different from the input. Like you have plenty of gaps in the input space of the compounds you might be feeding in, and already. Do we think that there's a lot missing from, the molecules we have information on, but two, are we able to at least generate, what fraction are we even not able to generate would be my question. How biased is this process? Yeah.
So regarding the way we generate molecules, I actually had a slide on that, but I did not, it was for the sake of time, I did not keep it. So there are basically using deep generative models, three main approaches which have been described, but it changes all the time because there is a lot of literature on this. So it's not it will change. But basically, the variational autoencoders was the first approach published by Duvneau in September 16, and we started with that approach. We decided to go away from that because we thought there were too many issues with regards to the quality of the molecules generated. And right now we have moved back to using another approach which is based on long short-term memory algorithms, where it's basically text generation, since you can encode a molecule as a SMICE, which is a string of characters with a syntax and a grammar. So we have more bet right now on this technology, and this is what we use to when in our projects. we still follow actively the literature on autoencoders, and actually we are quite interested in mixing chemical and biological characteristics as a way potentially to improve some of the aspects. But regarding the molecule generation, we get very good performance with the LSTM approach, and what is really nice is that it can be coupled with by reinforcement learning, as I explained, with any type of scoring system which does not have to be included in the latent space. So it gives you a lot of flexibility when applying to a concrete drug discovery project. Regarding the second question, it's an open question, and I don't have the answer. We are not academic people. We work for clients. We try to solve concrete problems that they meet in their drug discovery objectives. And here, in the project we saw with Servier, it was, it was a well-defined chemical space. I couldn't show you the diversity with the molecules, but basically, all molecules pretty much look alike with some diversity. And our objective is really to find optimal compounds with optimal profiles within that chemical space. So we don't have to solve the question of how much areas of the chemical space are really unknown, and can we generate molecules into that. When we work in scaffold hopping, it's a slightly different business. But again, I think we pursue very pragmatic goals, find new hits. We need them to be new, novel, patentable with FTO. We have succeeded into doing that, at least in silico, and we expect the experimental results quite soon with one of our clients. But we are not really yet into mapping the chemical space and identifying at a macro level unknown pockets where it might be good to explore.
I guess the other way to look would be, how often are these molecules things that there's information out there versus things that nobody's seen? Or you mentioned the retrosynth platform. How often you spit out things maybe that your retrosynthesis doesn't know how to make or something like this? I mean, these could be...
Yeah, it might be interesting actually to couple both approaches, because it's true that with retrosynthesis we build on existing knowledge, so it's only reactions, rules which have been previously described in the literature, and it cannot invent new reactions which have not yet been discovered by synthetic chemists. So that's the limitation of this approach. Yeah.
Thank you.